My absolute pleasure to introduce Dr. Lisa Johnson. Lisa is an assistant professor at the Feinberg School of Medicine at Northwestern University, Institute for Innovations in Developmental Science. She also affiliated with the Northwestern Weinberg College of Arts and Sciences. There, she participates in the interdepartmental neuroscience program, cognitive science, and clinical psychology program as a mentor. So she has a lot of hats within the institute. She graduated with a BA from psychology in the University of Chicago, got a PhD in cognitive neuroscience at the University of California, Berkeley. And she directs a dynamic brain laboratory where they focus on studying the brain dynamics, underlying information encoding, retrieval, and use across the human lifespan, from across levels of organization, from neurons to large-scale networks. In her work, she's focusing on studying the neural basis of memory, combining methods from cognitive psychology with an impressive range of cognitive neuroscience methods, including both invasive and non-invasive EEG, electrical stimulation, structural neuroimaging, and eye tracking. I like to call her my partner in crime in pioneering developmental, iEEG, and we'll hear about this and part of her other work that has been published in top-tier journals and really provides insights on the basic science of memory and brain development, as well as translational impacts more broadly. So join me in welcoming Lisa to CBR.
Awesome. Thanks for having me. Thank you, Noah, for that introduction. I think I will just dive in. We'll have time for questions. I expect this to take probably 40 minutes, so feel free to stop and ask. So I'm going to be really talking about a few studies that all get at the big question of how human brain development shapes memory formation. So we're going to all start on the same page here. What does human brain activity look like? So this is an electrical signal from an electrode placed directly on the visual or occipital cortex of a neurosurgical patient. It shows periodic fluctuations in excitability when the patient is looking at a blank screen. This green shading indicates that a visual stimulus has appeared on the screen. As you can see for yourself, these periodic fluctuations change in real time based on what the patient is looking at. Indeed, brain rhythms are ubiquitous, and lack of brain rhythms in the EEG signifies brain death, usually. So the electrical signal holds a ton of information, which we break down into components that fluctuate at different frequencies. The occipital signal you just saw fluctuated or oscillated at an alpha frequency, that is approximately 10 times per second or 10 hertz. The amplitude or power of the alpha response changed with the presentation of a visual stimulus. And in addition, a faster, higher frequency signal appeared and disappeared together with the visual stimulus. Frequency and amplitude are two characteristics of the signal. The third, which I'll talk about today, is phase, or the specific point along the rhythmic wave, measured in degrees along a circle. Whereas amplitude indexes signal overall power, phase indexes signal timing within a cycle. So two signals I will primarily focus on today are the high-frequency broadband activity, which measures local activity, and it is a proxy for measuring neuronal population activity. The other are brain rhythms, which coordinate local neuronal spiking and interregional connectivity. From these data, together, we study how the human brain encodes, maintains, and retrieves information, and how these processes vary by age across the human life. So brain rhythms change considerably during development. This makes understanding their development fundamental to understanding the development of the brain. Since the first human EEG in the 1920s, researchers have focused on alpha rhythms, which dominate the scalp EEG signal, especially during awake rest. Alpha rhythms emerge during infancy and exhibit rhythmic waveform patterns around 3 to 6 hertz within a few months. These rhythms then speed up and reach the canonical 10 hertz frequency or adult-like levels around age 12. This paper is from 1939. And the field remains in agreement. For comparison, on the right is a figure from a much more recent meta-analysis. Simply put, alpha rhythms emerge in infancy, they speed up across childhood, and they reach adult-like levels by adolescence. And these changes in brain rhythms mirror changes in brain structure. You're looking at here is a chart showing longitudinal data from over 100,000 individual MRI scans. In broad strokes, it shows that the volume and thickness of the brain's overall gray matter peaks in childhood and then gradually declines with advancing age across adulthood. Alpha rhythms seem to reach adult-like levels by the end of this period. By contrast, the volume of the brain's interregional white matter gradually increases across childhood and peaks in young adulthood. These changes in brain structure mirror changes in cognitive behaviors, including memory, attention, and cognitive control. These thinking behaviors mature over a protracted trajectory into young adulthood, and in most eventually decline even in healthy aging. So we really examine development across broad age ranges to study the neurophysiology of these thinking behaviors and to understand why they so often go awry in a slew of disorders we may face in our lifetime. To do this, we use parallel invasive and non-invasive electrophysiology to track brain activity in real time, associating human brain function, brain structure, and behavioral outcomes with high spatial and temporal precision. Today, I'm going to focus on our latest work tackling the how of episodic memory using invasive or intracranial EEG and human development. My colleagues and I, when I say that, I really mean Noah, have argued that such innovative research approaches will allow us to derive mechanistic explanations of the brain basis of behavior, a primary goal of cognitive neuroscience, and a critical component of any translation science. And we are very privileged to work with surgical epilepsy patients undergoing intracranial EEG monitoring. Patients, including pediatric patients, typically spend a few days in the hospital for seizure monitoring. During this time, we show up with a pop-up lab, run experiments, and then analyze the data from healthy tissue outside the seizure focus. I'm happy to talk more offline and more about how that method really works. But we do all this because these data provide unprecedented access to the human brain, affording both millisecond temporal resolution and centimeter spatial resolution. The application of intracranial data to understand human cognition has risen dramatically in popularity over the last decade, and our group is really leading efforts to apply it to understand the developing human brain. So to study memory development in neurosurgical patients, we used an established scene recognition task. In this task, 40 images of scenes are presented in the study or encoding block. During study, patients indicate whether each scene is indoor or outdoor. That's not intended to be a test. It promotes depth of encoding and then ensures that we as researchers can go back to the data and know which scenes were actually attended to. Patients are then tested on all of the studied scenes, intermixed with new scenes as foils in a recognition test. And as I already said, but I will reiterate, we always limit analyses to scenes that were properly attended to in the study block, as indicated by a correct indoor-outdoor response. At this point, we have data from over 130 patients, as shown in red. Importantly, memory performance improved in our patients from about age 5 to 30, and these developmental gains are comparable to those of a large cohort of non-clinical controls. This critical point provides some evidence that our data can generalize to the broader population. And of course, this pattern of behavioral gains into young adulthood mirrors the development of the brain. So using T1 MRIs obtained as part of surgical planning, we were also able to confirm that global gray matter volume decreases by age in our sample from childhood through adulthood. This point provides converging evidence that our patient data can generalize to the population. And diving right in, the age range of our first study, which is about 5 to 20 years, is marked by decreasing gray matter volume and increasing white matter volume. Specifically, adolescence is marked by the development of association brain regions, including but not limited to the prefrontal cortex. Association brain regions mature along a more protracted trajectory than sensory motor regions, consistent with the development of executive function or thinking behaviors in adulthood. Decreased gray matter volume specifically is posited to reflect neuronal pruning and thus support developmental gains in the efficiency of information processing. And indeed, in our first publication from this data set, which I cannot believe was 7 years ago now, we showed that earlier high-frequency broadband responses, which again are a measure of local neuronal population activity, within the prefrontal cortex partially explained developmental gains in memory performance over the age range of about 5 to 20. I will orient you to this plot, which shows stacked single trial power. So each one of these lines is one trial during which they were viewing a scene and saying indoor or outdoor. These recordings were all obtained from the inferior frontal gyrus, as indicated by those little dots over IFG. Each dot represents an electrode from a subset of patients with coverage of the inferior frontal gyrus. And these trials are stacked in order by the behavioral response time, with the curved line representing the onset of the indoor-outdoor response to each scene. So putting this together, yellow indicates significant increases over the pre-stimulus baseline. And you can see a pretty reliable increase in yellow preceding the behavioral output here. For comparison, if this were the motor cortex, it would be a beautiful line, basically on top of the behavioral output. And we found here that earlier inferior frontal gyrus activity during the study or encoding block predicted developmental gains in memory performance at subsequent times. So these data, although it's a small sample, provide initial evidence linking regional prefrontal cortex maturation to memory development with the temporal and spatial precision necessary to actually investigate processing efficiency in real time. In a much more recent publication, we found that behavioral gains in memory also mirrored the protracted functional development of the brain. So I'm going to switch gears a little bit here to slower brain rhythms. Shown here are intracranial recordings of single trials from the parahippocampal gyrus of a 6-year-old and a 19-year-old. So each one of these is a single trial. And on the left is the raw data, and on the right is the raw data filtered at the two most dominant oscillating or rhythmic frequencies. Importantly, these rhythms were prominent around 3 Hertz and 7 Hertz, which are near the edges of the theta band. Strikingly, we found that these theta rhythms separated into sub-bands within key regions supporting memory. Here, the parahippocampal and prefrontal cortices across development. So we discovered that slower rhythms got slower and faster rhythms got faster from age 5 to 21. And these results revealed for the first time that brain development is associated with the separation of the theta band from childhood into adulthood. Second, we found that the parahippocampal and prefrontal cortices were synchronized at both slow and fast theta frequencies during successful memory formation. And in both of these functional networks, connectivity was strongest in our top performing adolescents. So these results reveal that memory development is associated with the strengthening of multiple functional connectivity mechanisms. Third, we related these functional mechanisms to the structural development of the brain. So during development, as I said, concomitant with decreased gray matter volume is increased volume in white matter tracts supporting myriad cortico-cortico and cortico-hippocampal connections. White matter volume again peaks in young adulthood. And here on the right, I am highlighting the 2 major white matter tracts which connect the parahippocampal and prefrontal cortices. In pink is the cingulum, kind of long posterior route, and in light blue is the uncinate, or the short posterior anterior route. These 2 tracts I highlight because they are plausible structural connectivity candidates underlying the functional connectivity effects we observe. And indeed, we found that the strength of the cingulum tract, so that's the long posterior route, connecting parahippocampal and prefrontal cortices, itself predicted developmental gains in memory. So we have these mirrored effects in the functional data as well as the structural data, which we finally then just correlated to find that the strength of the cingulum tract was related to the strengthening of both functional connectivity mechanisms. This is really cool because it provides the first ever demonstration linking the structural and functional development of the brain with high resolution to the development of memory and furthermore starts to raise the issue of a potential topic area of multiplexing, in which two things are happening at once in the same place. And I think, and the rest of the data I'm going to show you today, I hope points to this as well, that part of what happens in brain development that potentially allows us to have these complex behaviors is our brain's ability to kind of juggle multiple things at once. So putting the pieces together, using the tools of intracranial EEG and human development, this research begins to reveal that key memory regions interact via multiple, increasingly dissociable mechanisms as memory improves from childhood into young adulthood. Medial temporal and prefrontal cortices interact via distinct rhythmic theta mechanisms during memory formation. These rhythmic mechanisms separate across development, and both are related to the structural integrity of the cingulum. So this tells us something fundamental about how the development of the human brain supports the development of memory and how memory becomes what it is. And these findings begin to uncover and to sort of further raise the intriguing possibility that the development of cognition broadly, including but not limited to memory, may relate to the development of these multiplexed rhythmic networks. Sort of like, as I said, the brain's ability to multitask. And likewise, neurological and neuropsychiatric disease, as well as neurodegenerative disease, may break down these rhythmic networks. These are major implications of our work and, of course, areas of future research. All right, the next study, all from the same task, focuses on late adolescence and young adulthood. And this period of late adolescence, young adulthood is associated with really achieving peak cognitive performance. So it seems to be a sweet spot of decreasing gray matter volume and increasing white matter volume. And this study is led by our postdoc, Adam Dede. He designed a very clever, albeit simple method, rather clever because it's so simple, method to examine dynamic brain activity and brain synchrony within single trials, with the goal of delineating how the frontotemporal network, so prefrontal parahippocampal connections, among others, encodes and retrieves memories in real time. So to do this, he analyzed data from 36 adolescents and young adults, which were selected based on electrodes sampling any of 5 frontotemporal regions of interest. And he first focused on the high-frequency broadband response, which again is a measure of local neuronal population activity. On a trial-by-trial basis, he isolated the latency of the peak high-frequency broadband activity. That is the moment of maximal neuronal population activity, bounded any time between the onset of the scene at time 0 and the behavioral indoor-outdoor response, which here on this schematic is shown in red. He then computed theta functional synchrony between regions, time-locked to the high-frequency broadband peak as a function of memory success. So here we're looking at scenes as a function of whether they were later remembered or later forgotten. Recognition test. This method isolates memory effects while controlling for inter-individual and inter-trial variability and internally generated information processing, and thus illuminates the dynamics of memory encoding and retrieval in real time. And there are a lot of findings in this paper, and I am going to actually encourage everybody to go to our preprint. Some QR code at the bottom, but briefly, I'm going to summarize some of the main points here. So what you're looking at here is a schematic of kind of all of the connectivity effects put into one, so we can really look at temporally constrained brain networks. So on the left here is the dynamic synchrony profile of successful encoding, time-locked to the internally generated high-frequency broadband peak within single trials. By comparison, on the right is aligned to the externally presented scene. 2 major points. First, regions of the medial temporal lobe are synchronized in the slow theta band, regardless of how the data are aligned. By contrast, synchrony between medial temporal and frontal regions depends on how the data are aligned. During memory retrieval, to complicate things, regions of the medial temporal lobe are desynchronized, but again, regardless of how the data are aligned. And again, synchrony between medial temporal and frontal regions varies depending on how the data are aligned. Across the board, the anterior cingulate, which I think we would all, if asked, agree participates in high-level cognition, seems only to be engaged with the frontotemporal memory network when the data are aligned to the internally generated high-frequency broadband peak. There's a lot more to unpack here. But the bottom line is that the results of trial-by-trial analysis challenge a simplified story of frontotemporal interactions we know based on trial averaging to an externally presented stimulus, and specifically link anterior cingulate engagement to internally generated information processing. That is all to say that over the course of even just 3 seconds of looking at a scene, a lot of what happens to make memory successful is driven by your internal processing, which can happen at any point during that 3 seconds. And some of the guiding regions there seem to be more frontal region, including the anterior cingulate and regions more anterior to that, whereas the medial temporal lobe, less about internally generated information processing, responds to the frontal lobe, also responds to the onset of the scene. Based on these results, to get one more step complicated, Adam hypothesized that memory success would actually depend on precise dynamic. That is, specific connections at specific. To test this hypothesis, he took these dynamic synchrony data and subjected them to 3 different graph theoretical metrics in order to examine connectivity across the entire frontotemporal network within the high-frequency peak broadband aligned data. And the results were striking. So this is just one representative participant, but you can really see that it is indeed specific connections that seem to dictate whether or not information is successfully encoded into memory. On the left is a successfully remembered scene, and on the right, an encoding failure. These results further challenge dominant models that say that more connectivity is always better. And more to come. This paper's under review right now, and we are really excited to see where this work will go. All right. Finally, for the second half of the talk, I'm stoked to introduce a relatively new line of work mapping the maturation of the human brain from about age 5 to 55 years. Basically, to lay the foundation to address big questions about how the human brain develops into the information superprocessor that we associate with healthy young adulthood, and how aging and disease can break down neurophysiology and impair cognition. This work is spearheaded by our postdoc, Zach Cross, who is visiting me here. So feel free to ask him things too for visiting with me. All right. So first, inspired by our discovery that the theta band separates from age 5 to 20, we are asking, does age-related variability in theta rhythms plateau? We need to look above age 20 to address this question. Intracranial EEG is typically performed in patients up to about 50, 60 years. So we can do that. Is age-related variability in theta rhythms specific to task engagement? Right, everything I've shown you was during the encoding of scenes. We can compare task-free or resting state data to address this question. No problem. Does age-related variability in theta rhythms predict memory outcomes? So not only is it happening, but in reference to our specific question, how important is it? Memory improves up to about age 30 with marked inter-individual variability. Predicting such inter-individual variability is critical to be able to understand healthy and disordered brain development. Finally, what about the background aperiodic 1/f-like activity? So in parallel, we are asking all of these same questions of the aperiodic ubiquitous background signal. What does that mean? Here's a schematic. Zach analyzed data from over 100 patients to begin tackling these questions, about 60-40 male-female. This exceptionally large sample for intracranial research, this sample is bananas. Basically, this exceptionally large sample provided beautiful coverage of all cortical regions, allowing him to really analyze inter-individual variability within localized brain regions. And simply put, he applied fancy math to separate the power spectrum of each electrode in order to isolate rhythmic peaks Those are the bumps in the power spectrum, like that, that indicate theta and alpha rhythms from aperiodic activity, which is measured by the slope and offset of the background line without the bumps. Basically, this line, and then we've got these. First, we found that global, so across all 5,000 plus electrodes, slow and fast theta rhythms do in fact separate in frequency from childhood into young adulthood. So that was really exciting to see after our initial finding with an N of about 20. We're now finding it in a big N and across a longer age range. We found that global theta band separation peaks around late adolescence, young adulthood. Of course, is that moment of peak cognitive performance where your brain seems to really be in a sweet spot. Separation then decreases gradually with advancing age across adulthood, largely driven by the slowing of fast theta rhythms with advancing age. Next, it's just a visual summary of mean, peak, slow, and fast theta frequencies across all brain regions. If you squint, you can kind of see a posterior to anterior gradient in the speed of slow theta rhythms, the opposite in fast theta rhythms. We don't know what that means yet, but it does really show us the spatial specificity of this effect. Next, focusing on some key regions supporting memory. So these are the same ones I keep coming back to, prefrontal and medial temporal. He found that theta rhythms vary by age differently during task engagement, so memory encoding, and task-free rest. Intriguingly, the differences between task engagement and rest consistently reverses in adolescence, consistent with the protracted development of executive function or thinking behaviors. And importantly, he did find that age-related slowing of slow theta rhythms within the prefrontal cortex predicted age-related improvement in memory outcomes. So all of this stuff really matters for the construct that we're looking at. Basically, when slow theta frequencies do not slow down with age, memory suffers. By contrast, slow theta frequencies in the hippocampus predict memory outcomes independent of age. Here, when slow theta frequencies are too slow, memory seems to suffer. And in both regions, the effect was specific to theta rhythms during memory encoding, suggesting relationships specific to task engagement. And these effects mirror developmental changes in regional brain structure. So whereas association regions, such as the prefrontal cortex, continue to develop into adulthood, mirroring the development of thinking behaviors into adulthood, total hippocampal volume is largely stable from childhood. Right. And finally, as I said, he asked the same questions of aperiodic activity, starting with a summary of mean aperiodic slopes across all brain regions. If you squint, you can see a posterior lateral to anterior medial gradient in the flattening of aperiodic slopes. This is especially intriguing because flatter slopes are posited to reflect increased excitation relative to inhibition. Next, we found that global aperiodic slopes flatten from childhood into young adulthood, suggesting increased excitation with development. We found more flattening in sensory motor than association cortices. Surprisingly, however, the age of maximal flattening was the same across sensory motor and association cortices. This result challenges models of early sensory motor development based on brain structure alone. We found that aperiodic slopes then steepen with advancing age across adulthood, largely driven by the steepening of sensory motor slopes in middle adulthood. And mirroring analyses here. Next, he found that aperiodic slopes in the prefrontal cortex vary by age differently during task engagement and task-free rest. Steeper slopes suggesting increased inhibition during memory encoding consistently emerge in adolescence, again consistent with the protracted development of executive function or thinking behaviors. And this occurs against a backdrop of flatter slopes by age, beginning to point to a Goldilocks phenomenon. Sort of sweet spot again. And importantly, he found that the age-related flattening of aperiodic slopes in the prefrontal cortex predicted age-related improvement in memory outcomes. I think you might be starting to see a pattern here. Here, when aperiodic slopes are too steep in adolescence, memory suffers. By contrast, aperiodic slopes in the fusiform gyrus, a region adjacent to the medial temporal lobe that is involved in high-level visual processing, shows the opposite pattern. So here, when aperiodic slopes are too flat in adolescence, memory suffers. And in both regions, the effect was specific to aperiodic activity during memory encoding, suggesting relationships specific to task engagement. So in conclusion, using the tools of intracranial EEG and human development, this research begins to point to regionally precise Goldilocks phenomena and link age-related variability in prefrontal cortex neurophysiology to age-related variability in memory outcomes. Adults generally exhibit better memory than children, and age-related improvements in memory are associated with flatter aperiodic slopes, reflecting increased excitation, and slower slow theta brain rhythms, potentially related to increased capacity for information processing. Memory suffers when neurophysiology deviates from the aperiodics. These findings tell us more about how the development of the brain supports the development of memory, how the human brain develops into the information superprocessor we associate with healthy young adulthood, and how aging and disease can break down neurophysiology or cognition. Finish our outline. A few minutes early, and I will just briefly thank everybody. So this is more than just our lab here, but some fun outing we had when the weather was beautiful over the summer. And a huge thank you to Noah Offen for inviting me to do this, and also for being my partner in crime for 9 years, 8, been a while. My other advisor and PhD advisor, Bob Knight, who's a major collaborator to enable a lot of this work for us, and Zach and Adam, whose work I really highlighted today. Thank you.
Thank you. And I'm going to open the floor for any questions that people might have brewing. Yes, Kendra, raise their hand.
I'll just ask a clarifying question. So the aperiodic slopes you're talking about, is that related to 1/f scaling that you mentioned earlier?
So we call it 1/f like because that is the shape it takes. Like it follows the power scaling law.
Okay.
But it is not a literal representation.
Okay.
Basically, if your slope doesn't look like that, there's something kind of wrong with it. That's everything follows the power scaling.
Okay.
Excuse me, 2 questions. One of them is related to the analysis you were doing that was predicated on looking for the peak of high frequency activity. Was that high or low gamma?
That was high gamma, so bounded either 70 or 80 to 150 hertz.
So what is it that makes you think that the peak is special? If I, as a theory of brain function, why should point in time when that integrated power reaches that peak in a trial be so important? What's the theory behind that?
So that's a good question. The theoretical motivation behind that is that high frequency broadband activity, it is not a perfect representation of neuronal population activity. It is a correlate for a proxy measure. It basically measures neuronal population activity a little more. But what it is a really reliable, temporally precise marker of local, regional, just activity, neuronal firing, and a little more. And so finding the peak is that moment of the most engagement of that tissue in whatever you're doing. And so it's a method by which we can actually look in real time at what is the brain doing when this region is maximally.
I'm just thinking a lot goes into that in terms of the local dynamics. So it may not be the point at which you get maximum engagement, meaning presumably maximum synchrony of synaptic potential, something like that. That may not be the point at which the brain is computationally at its maximum.
Sure, that is,
That's, I guess, what I'm trying to.
yeah, I guess there's an assumption that more neuronal firing means more engagement. Because we can't prove that by simply finding the point of more neuronal firing, but what we are finding is the point of the most neuronal firing.
Second quick question, which is in no way specific to your work, we all have the same problem, is that essentially all of this work is correlation. And it doesn't license any kind of causal conclusion about which, if any, of these patterns of brain activity are actually causally related to the behavioral phenomenon study. Have you thought about doing disruptive stimulation through the same electrodes that you're recording from?
Yeah, so that is, to unpack that a little bit, right? This really, all of this work is observational, absolutely, and I do not claim that it's mine. So looking at things like, to what extent does measure X, be it theta frequency, aperiodic slope, steepness, whatever, predict memory outcomes, that is a multiple regression. It's just fancy correlation. It does not say that you need this in order to have good memory outcomes. But it starts to give us one piece of the puzzle, saying this is probably helpful. And then the parallel line of work ideally would allow us to say, disrupt that signal and see if memory falls apart. That would allow us to causally attach something to it. That is doable in intracranial patients, as you know. Any electrode that measures current can deliver current as well. It is currently, I shouldn't have used the word current there presently, only really performed for research in adults. So it is not really a way to get at these potential causality of age-related variability, because we really can't do it in kids without a clinical rationale.
The reason being, what has anyone tried with an IRB to see whether that would go through?
A surgeon probably could get away with it. I would need to collaborate with a surgeon to make it happen. And it's certainly something that we have talked about. And it would be extremely cool. And I think it's not a closed door forever. It's one we haven't gotten to.
Okay.
But what I can say is, in parallel work with, say, scalp EEG and brain stimulation, it doesn't get you that kind of spatial precision, but you absolutely can disrupt, say, theta rhythm and see what happens to memory. And the results are not super reliable with that kind of technique, but just if you go to the literature and check them out. But with something really focused, like here, we have these data that would maybe provide a really strong expectation of exactly what we're looking at. Like, okay, this person is 35, therefore we expect slow theta to be within this range. We can try to explicitly tap that slow theta for their age range and then see how their memory responds. We can try to target iEEG, but yeah, this is sort of always an issue. And then it gets even more philosophical. Can you really use the word mechanism, causality? And I can only ask that. I can't. But yeah, this is part of a big sort of thought of what would be a bunch of parallel lines of work to really get it, like the causal mechanism by which memory changes by age, of which we got a small part of that. Correlational mechanism by which memory varies by age.
So could I just follow up on that, just to clarify. So is that specific to an experimental context, or are you saying that in the pediatric samples they're not doing any stimulation mapping either?
Oh no, they do clinical mapping, but that's for clinical purposes. To do it explicitly for research is going to be complicated with an IRB.
But you can get
Not impossible.
You can get the data out of that.
Yeah, but it wouldn't be during our task. Right.
Even at rest, you can get the data out and look at how the productivity change.
Yeah. Sure. In fact, there's a very cool, Dora Hermes' group has made open source a big developmental data set of just looking at the single pulse stimulation, cortico evoked potentials at rest. And again, I'm like, when we talk about those data, I always wish it were somehow aligned with the task so we could start to get at these questions of memory. But yeah, that would be an area to look at. Sure.
Thank you so much for the wonderful talk. I have a question. It looks like the kids it didn't show as good as memory as in adults. So I was wondering whether, how in research is it tackle the possibility that the kids, they may didn't really learn the content or learn the stimuli as well as the adults. So probably like when they didn't really show the good memory, it's possible that they didn't really like good encoding or good perception rather than it was possible, rather than there's problems with the memory.
I think it's more than possible. We know part of the reason that kids' memories are not as good as adults is due to encoding.
Okay, but if we want to have a clearer understanding of where or why or how this happens, is there any possible solution to that?
So I guess what I'm saying is that that's a main thing that I was just showing is part of how that happens.
Can
It's-
repeat the question for that?
Yeah, what was it? It was, can you look at children to understand how that happens? Why encode, like how encoding would vary by age such that memory would improve. And there's a lot more to look at. I focused only on studies that looked at the prefrontal cortex and the medial temporal lobe. But yeah, so putting some of this work together, it's that the frequency of theta rhythms varies by age. The strength of connectivity within each of those theta rhythms varies by age. Each of these are independently contributing to memory improvements by age. In addition, we see brain structure here, the cingulate tract, also predicting memory improvements by age. There's a whole lot going on, but all of these things are related to, in some way, age-related variability and functional connectivity during memory encoding. And that is just part of the puzzle. We've barely, scratched the surface of, say, memory retrieval, where we know there's certainly developmental variability in retrieval processes as well. And I did not show anything from like the back half of the brain today, which also participates. It's not all prefrontal cortex.
Do we have any questions on the chat?
We do not have any questions.
Last chance. So with that, I'll just thank Lisa again for a really enlightening talk, and thank you all for your attention.